enzymes are classified into 7 main classes based on the types of reactions they catalyze. Using the first letter of each class of enzyme, let's use the rhyme "Oh, the hungry lion is large too" to easily remember the 7 classifications of enzymes. O stands for the Oxidoreductase, D stands for Transferases, hungry stands for Hydrolases, lion stands for Lyases, is stands for Isomerases, large stands for Ligases, and 2 stands for Translocases. Therefore, with this rhyme, you should easily remember your classes of enzymes. Now the Oxidoreductases. This class of enzymes control oxidation-reduction reactions. They catalyze the transfer of oxygen, hydrogen atoms, or electrons from one molecule to another. In this reaction, electron is transferred from molecule A to molecule B, catalyzed by oxidoreductase enzyme. Therefore, molecule A loses an electron, and that is an oxidation reaction. Molecule B accepts an electron, and that is called a reduction reaction. The electron donor, molecule A, is termed a reductant, and the electron acceptor, molecule B, is termed an oxidant. Now, here is a tip. Most oxidoreductase enzymes in biochemical pathways are termed dehydrogenases. Other subclasses include the oxidases, the reductases, peroxidases, etc., all distinguished by their different electron acceptors. Succinate dehydrogenase, an example of an oxidoreductase, catalyzes the conversion of succinate to fumarate. In this reaction, FAD coenzyme accepts 2 hydrogen atoms from succinate and becomes reduced to FADH2 as succinate is oxidized to form fumarate. You can think of oxidoreductases as energy managers of the cell. Now the transferases. This class of enzymes transfer chemical groups from one molecule to another. Transferases move a variety of chemical groups such as the amino group, the methyl group, phosphate group, etc. Alanine aminotransferase is an example of a transferase enzyme, and with pyridoxal phosphate serving as a cofactor for this enzyme catalyzes the reversible transfer of amino group from alanine to alpha-ketoglutarate, resulting in the formation of pyruvate and glutamate. You can think of transferases as molecular delivery agents. Now the hydrolases. This class of enzymes use water to break down large molecules into simpler units. They break down proteins, fat and carbohydrates into amino acids, fatty acids, and sugars. Examples of hydrolases include the proteases, the lipases, phosphatases, nucleases, glycosidases, etc. Now, protease, which is a type of digestive enzyme, cleaves the peptide bond in proteins, resulting in smaller peptides or individual amino acids. You can think of hydrolases as the scissors of the cell. Now the lyases. This class of enzymes catalyze the breaking of a chemical bond without involving the use of water, unlike hydrolases. The resulting product involves the formation of a double bond or addition of a group to a double bond. Examples of lyases include aldolases, decarboxylases, dehydratases, etc. Now, the various types of lyases are distinguished based on different bond cleavage, which could be a carbon-to-carbon bond. It could also be a carbon-to-oxygen bond, a carbon-to-nitrogen bond, amongst others. Now, given this example, pyruvate decarboxylase, a lyase enzyme, breaks a carbon-to-carbon bond in pyruvate, removes a carbon dioxide molecule, resulting in the formation of acetaldehyde. Think of lyases as chemical quarters. Now the isomerases. This group of enzymes catalyze the structural rearrangement of molecules, converting one isomer to another. They do not change the overall molecular formula of the molecule. An example of isomerases include triose phosphate isomerase, alanine racemase, glucose isomerase, amongst others. Now, given this example of an isomerase catalyzed reaction, the enzyme glucose isomerase catalyzes the reversible conversion of D-glucose to D-fructose. You can think of isomerases as molecular reshapers. Then the ligases. This group of enzymes, they join 2 molecules together using ATP. Now, examples of ligases include the DNA ligase, succinyl-CoA synthetase, pyruvate carboxylase, amongst others. Now think of Lyases as molecular glue. Now with this reaction, with the utilization of ATP, DNA ligase connects the 3' hydroxyl end of one nucleotide with the 5' phosphate end of another nucleotide. The enzymatic activity of DNA ligase results in the formation of a strong phosphodiester bond, which acts as a molecular glue and joins the two nucleotides together. Then, the 7th class of enzymes is the translocases. These enzymes facilitate the movement of molecules or ions across membranes. And examples of translocases include mitochondrial translocase, carnitine acyl carnitine translocase, ornithine translocase, etc. Now you can think of translocases as the movers of the cell. This figure illustrates the catalytic activity of a translocase enzyme in moving molecules across a cell membrane at the expense of ATP, which drives the activity of the enzyme. Now the summary of our lesson today is that there are 7 main classes of enzymes, and using the memory trick, "Oh, the hungry lion is large too," you should easily remember your enzyme classifications. Oxidoreductases catalyze the redox reactions. The transferases move functional groups between molecules. The hydrolases breaks bond using water. The Lyases removes or adds group without water, often forming double bonds. The Isomerases rearrange atoms within a molecule. The Ligases joins molecules using ATP. And the Translocases moves molecules or ions across cell membrane. If you found this lesson helpful, please drop a like, leave a comment, and subscribe for more simplified biochemistry.